Have you ever stopped to think what a spider web, your immune system, and the muscles that let you walk all have in common? Well, the answer is proteins. They are, without a doubt, life's most incredible and versatile little machines. And today, we are going to unfold the secrets of the molecular origami that makes it all possible. So let's dive right in and talk about just how essential these molecular workhorses are to, well, literally every living thing. I mean, just think about the sheer range here. Proteins are what gives spider silk its incredible strength. They're the antibodies that act like your body's personal security team. They're hormones carrying critical messages all over the place. They're actin and myosin, the fibers that literally pull your muscles together. And of course, they're the enzymes breaking down your food. They do just about everything.
And it's absolutely no exaggeration to say that proteins are everywhere. They are the most common large molecules in nature, and you'll find them in every single cell of every living organism. They are truly the substance of life itself.
Okay, but here is the part that is just wild. All of that amazing variety, from a spider web to an antibody, is built from the exact same universal toolkit of just 20 different building blocks. That's it, just 20. We call them amino acids. And that brings us to the big puzzle, right? How on earth is that possible? How can life create this endless universe of molecular machines from such a simple 20-piece set? Well, the answer lies in a beautiful process, a kind of molecular origami. To really get how this works, we've got to get to know our toolkit, those 20 amazing amino acids. So every single amino acid shares the same basic backbone. You can kind of think of it like a standard Lego brick. But attached to each one is a special, variable part called the R group. And that R group is what makes each of the 20 bricks unique. It's like the special connector on top of the Lego that decides how it's going to fit in with all the others. Now, these R groups come in about 5 main flavors. You've got some that are nonpolar, some that are polar, and others that carry either a positive or a negative electric charge. These different chemical properties, well, they're the secret sauce to everything that's about to happen. So why do these flavors matter so much? It's actually pretty intuitive. The nonpolar ones are hydrophobic. They're afraid of water. And since our cells are mostly water, these guys will do anything to hide on the inside of the protein. The polar groups are the exact opposite. They're hydrophilic. They love water, so they're happy to hang out on the outside. And then the charged groups can form these strong bonds with each other, kind of like tiny little magnets that help lock the final structure in place. And all of this brings us to the main event, how a simple chain of these amino acids folds itself into a complex, functional, three-dimensional machine. This, right here, is life's molecular origami. This folding process happens in 4 distinct stages. First up, you have the primary structure. That's just the linear sequence of amino acids, one after another, like beads on a string. Then, parts of that string start to form simple local shapes, like coils or folded sheets. That's the secondary structure. After that, the entire chain scrunches up into its final, unique 3D shape. This is the all-important tertiary structure. And finally, some proteins are so complex, they need multiple folded chains to come together to form one giant machine. And that's the quaternary structure. Now, those 2 most common secondary shapes are the α-helix and the beta-pleated sheet. The α-helix is just what it sounds like, a spiral, kind of like a corkscrew, and it's held together by bonds within that same chain. You find the structure all over the place in proteins like hemoglobin. The beta-pleated sheet, on the other hand, is flatter, more like a fan or a folded piece of paper. And it's stabilized by bonds between different parts of the chain lying next to each other. This is the structure that gives silk its incredible strength. So what holds that final 3D shape together? It's all those R group properties we talked about earlier. The biggest driver is that hydrophobic effect, all the water-fearing bits burying themselves on the inside. Then you've got tons of weaker hydrogen bonds and those stronger ionic bonds, like little salt bridges between charged groups. And sometimes you get these super strong links called disulfide bonds that act like molecular staples, locking everything down tight. Okay, and now for the big payoff. Why have we gone through all this detail about folding and chemical properties? Because it all leads to what is arguably one of the most important principles in all of biology. So, the crucial point is this. It all begins with that primary structure, that specific one-dimensional sequence of the 20 building blocks. And that sequence, based purely on the chemical nature of those R groups,
automatically and precisely dictates how that chain is going to fold up into its final complex three-dimensional shape.
And that final, unique shape is what gives the protein its unique job. An antibody has a shape that's perfect for grabbing onto a virus. An enzyme has a shape that creates a perfect little pocket to break down a sugar molecule. The shape is everything. This simple, beautiful flow of information is the core idea you need to remember. The one-dimensional sequence determines the three-dimensional structure, which in turn determines the biological function. Say it with me. Sequence determines structure, and structure determines function. It's truly one of the most powerful and elegant rules in the natural world.
Which leaves us with a final, pretty mind-blowing thought. For decades, being able to predict a protein's 3D shape just from its sequence was one of the holy grails of science. But we're getting really, really good at it. So the question becomes, if we can truly master this molecular origami, if we can design proteins to fold into any shape we can imagine, what problems couldn't we solve?